So now what we're going to talk about is energy. Without energy, there would be no life on the planet. And as we think about stuff, life, these biologically important molecules, without getting into any theology at all, just staying scientific because this is a science class. If you want to talk theology with me, we'll talk about that all day long. We'll just do it after class. But when you look at the stuff that is present in the universe, we all came from stars. Stars and their burning energy created these heavier and heavier atoms that exploded out into the universe to produce the carbons and the oxygens and the nitrogens. And so we're a collection of stardust. And when you look at life in general on the planet and you look at all energy, all of our energy that we have comes from a star. And what is our star called? The sun. All energy that we know comes from the sun. And it's a great thing we have the sun, and we live a certain distance from the sun. They call the Goldilocks zone. That's what astronomers do when they're looking for planets around other stars. But all of that energy comes in is required for cells because we have to maintain a body temperature. We have to polymerize certain molecules. We wouldn't be here without the movement of a spermatozoa that fertilized an egg. Concentration gradients, charge separation, we're going to get to those shortly. But this is all energy that happens in all of your cells each and every day, and it all starts with the sun. And we have certain groups of organisms on the planet, autotrophs. Why do you think there's a green box with a P in it? Who are autotrophs? Plants, photosynthetic organisms. They capture the energy from the sun, those photons of light, and they convert that energy into chemical energy, and they store it in chemical bonds of molecules, not the least of which is our 6-carbon ring good friend called glucose. Then we have herbivores that come along. They eat the autotrophs. Then we come along, and you gotta like a good steak, right? So we get the energy from the herbivores, and we start this process over and over and over. But what I want to point out in this conversion process from autotrophs, herbivores, carnivores, decomposers, we don't transfer all the energy. Some of it is lost. So it's not a 100% efficient process. And when we say the energy is lost, it doesn't mean it disappears. It doesn't mean it's gone. Because we have two very fundamental laws of physics. And I know this is not a physics class, but this completely relates to looking at energy. The first and second law of thermodynamics. The first law, and you notice that I've got an underline here that says change. When you think of the first law, I want you to think of change. Because energy, by this very first law, it cannot be created and it cannot be destroyed. It can only be changed. So the energy we get from the sun, we change that. Plants change that. Animals eat the plants. They change it. We eat plants and animals. We change it. Some we don't get to use directly. Some is lost as heat. But the energy never goes away. It's simply transformed. Which always makes me curious. Because invariably, I always get this question. Because I go to church, but I'm a scientist. And a lot of people always ask me, how do you reconcile the two? Well, I always talk science because this is a science class, but this very first law, energy can't be created or destroyed. Well, where'd all the stuff come from? The first law says we shouldn't be here. Did it just always exist? I'm not trying to provide you answers. I just like asking questions. But that's a big one. Where did everything come from? When? Way back. So anyway, we'll leave that one alone because it's making my head hurt. First law, change. You only change energy. The second law. Now we mentioned entropy earlier. That's just a term I wanted you to be familiar with. And we're not going to spend a lot of time trying to really understand energy other than this disorganization in the universe. But since everything wants to become more disorganized, the second law says, we're going to lose energy and heat. As we transform energy, some is going to be lost in heat. You just have to. It's like when you transform energy, somebody gets a commission. Has anybody ever worked in food service and stuff, and you got to give a part of what you earned? Yeah. Have you ever had a job and you see FICA getting most of your check? Well, I guess it's kind of like this one. Yeah, you're going to get lost in taxes. So you can only change the energy, and it's not going to be a 100% efficient transformation. Some is going to be lost. But regardless, where does it all come from? Where does all of our energy come from? The sun. That's where it starts. So at some point, we have to begin to get down to our cellular level, our organelle level, and our molecular level. And so as we look at this illustration, understand what we're looking at here in the green box in photosynthesis. This is an organelle that you find in plants and some fungi. This is called a chloroplast. This is where photosynthesis takes place. So photosynthesis is where the energy of the sun is captured. You actually have two steps of photosynthesis. You have a capture stage of photosynthesis, and then that energy is used to take carbon dioxide and attach the carbons together and produce glucose. And that second stage is going to be our energy stored stage. Now, photosynthesis gets a lot more technical than that, but that's all we're going to do. Photosynthesis captures energy and it stores energy in chemical bonds of glucose. Now, once we come over to the animal side, in the animal side, we are going to take the glucose where the energy was stored, and we're going to transform that energy. We're going to transform that energy using oxygen. So this is aerobic, introducing some terms that we're going to put in a better picture and look at even closer probably on Wednesday. But we take the glucose and oxygen and we transform the energy. We take the energy out of the chemical bonds of glucose and we put that energy in the bonds of ATP, adenosine triphosphate. This is the same A that we see in our nucleic acids. We're just using it differently. So the bond of energy between the second and the third phosphate, where we get the triphosphate part, that's the energy that we're getting from those chemical bonds of glucose that originally came from the sun. You see that? So I like to say ATP, the energy currency of all cells. Every cell uses ATP. It's like your debit card. If you've got money in the bank, your debit card's ATP. You can get work done with it. You can buy stuff. But if your debit card's like mine, and there's no money in the bank, then my debit card is ADP. I don't have that third phosphate. I don't have the energy stored there. I guess I could use it, but I go to jail, right? No, it's not going to get much work done with my debit card. So ATP, this is the form in which we are going to use the energy to do everything that our body does. Muscles are going to contract. Nerves are going to send signals. Immune cells are going to crawl around. That's where we are getting the energy from the sun into ATP so that our cells can use it for everything they need to do. Now, for us to be able to do that, we have to understand some foundational principles of energy. And the two types of energy are potential and kinetic. And I think we're pretty good with that, so this is kind of review. A potential energy is a substance or an activity in which you have stored the energy, and once you release that energy, you can get work done. And so when you pull back a bow, because it's getting close to deer season, right? You pull back the bow, that bow has some potential energy in it. All you have to do is let go of that string, and you're going to convert that potential energy into kinetic energy and get movement. For your cells, some of that potential energy is going to be in concentration gradients on either side of a plasma membrane. We're going to see this a ton. And when you think of concentration gradients relative to cells and membranes, I want you to think of a hydroelectric dam. You dam up a river and the water stacks up on the lakeside. The water goes down on the riverside. Do we just leave it there so we can go water skiing and fishing? No, what did they use the dam for? Energy, to make electricity. How do they do that? The level of the water on the lakeside has a potential energy because of gravity. Gravity's wanting to pull it downhill. So when they open the gates and allow that water to come through the turbines, that the water then spins. I mean, this is like an old-fashioned water wheel. You've seen those in some of the old movies. It spins that turbine. That turbine is attached to a rheostat that produces electricity. So you turn the potential energy of the water building up to kinetic after you let it flow through. Your cells are going to build up concentration gradients of ions, and when you open a channel and they flow through, you're going to be able to accomplish work. So do you see the potential energy at the cell level? And again, compare it to what's happening at the lake. Chemical energy. This is another type of potential energy, because here's our 6-carbon glucose molecule. And it's showing these little yellow kind of star explosions because, again, the energy from the sun is what is stored between these bonds, between our atoms, that we then transform, capture, and use as ATP so that we can cash it in when we need to do things like work our muscles. And we refer to that as the chemical energy, the energy of those bonds. All of our atoms are held together in such a way that they all have a potential kind of energy that we call chemical energy. So we get glucose. Our cells break it down. We go through respiration, which is that transformation and capture of the glucose energy and making it into ATP. Then we use ATP so we can accomplish kinetic energy. Our heart beating, those muscles contracting, kinetic energy. Here we have hearing, those sound waves impacting on our tympanic membrane. What's another name for the tympanic membrane? Eardrum. That's going to be a type of kinetic energy, mechanical energy, because you have that vibration. We have light energy impacting on your retina. Your retina is going to convert those photons of light into chemical gradients that will signal your brain. And then, of course, we have those other electrical chemical signals that are going on in our nervous system. Our nerve cells, or neurons, have that type of electrical energy. Now, since I mentioned neurons, I wanted to show you this. This is our potential energy picture where we have sodium ions stacked up on one side of the membrane, much lower concentration on the other. This is how neurons work to send signals. When that nerve cell body receives a change of electrical signal, which means you need to send a signal down the line, these sodium channels open. Sodium flows from the outside of the cell to the inside of the cell. which changes the electrical distribution of those charges, which changes the voltage. And so this is a measure of the voltage change that happens, and that change flows down that part of our neuron called the axon, and it's illustrated graphically as this electrical wave that goes down the line. All that wave is a changing of your sodium gradients, and when we get to nervous system, we're going to see also a change of our potassium gradients. So do you see in this particular example, we started with a potential when the cell was at rest, and by allowing the kinetic energy of sodium moving, we can send a signal all the way down that cell. And that signal may go from your spinal cord to your big toe or back. So potential and kinetic. Now, to understand this conversion process, to understand the capture even in photosynthesis, which it's not the scope of our class, but we have to understand chemical reactions. Are most of you also in chemistry this semester? In one way, shape, or form, or you will be? So, with a chemical reaction, we start with certain molecules. We may call them the reactants. And as we undergo a chemical reaction, we're going to convert these reactants into some form of product. And typically, we're going to be breaking chemical bonds, causing the chemical bonds to rearrange, and we're going to form new chemicals at the end of the process. So under certain conditions, bonds can be broken, molecules rearrange, and we end up, when we started with hydrogen peroxide, now we end up with water and oxygen. So again, chemical reactions, breaking bonds, rearranging, reforming new molecules. Now, when we put it on paper, like in a chemistry class, we draw it out like this. 2 molecules of hydrogen peroxide yield 2 molecules of water and 2 molecules of oxygen. Now, when you write it out, everything has to balance. You're not going to lose atoms. You're not going to lose ions. And the important parts of these reactions are the arrow, which indicates, okay, which direction are we moving? The reactants, this is the stuff that's around before the chemical reaction started. The products, what happens after, and oxygen, the water is also a product. I should have colored that purple as well. And again, as you're trying to balance these out in chemistry class, here we have 4 atoms of hydrogen as a reactant. Here's our 4 atoms here. We've got 4 atoms of oxygen. We're going to put 2 of those atoms with our water, and there's 2 left over as simply O2. That is a good, balanced chemical equation. So we left off last time as we were talking about chemical reactions. So we had a very simple one to begin with. I like to start simple and build and get more complex. If you start out too complex and you don't understand, well, you miss the bus and you're never going to get back on. So as we look at our equation, we have 2 molecules of hydrogen peroxide. It undergoes a chemical process, and we're going to explain a little bit more when we get into some specific examples. But in this chemical reaction, the hydrogen peroxide breaks down into water and oxygen. Now remember, what you start with are called the reactants. What you finish with are the products, including water. But in a chemical reaction, the arrow can go in which direction? I heard somebody say either. The arrow can go in either direction, but it can also do one other thing. It can go in both. You see how those words are different? either sort of signifies a single direction, but if you do both at the same time, it is a reaction that is reversible. So that's just sort of a broad understanding of a chemical reaction. And when you do, in fact, get into chemistry class, you're going to have to balance out the equation by keeping up with the atoms. We're not going to do that kind of balancing and stuff. This is just, again, to introduce this concept to you, because as we get into using energy and utilizing energy, we have a much more complex equation. And even though it looks really bad, let's kind of step through it just a little bit. When you see an equation that says CH2O, what category of macromolecule are you automatically thinking about? Carbohydrates. That is the basic formula for a carbohydrate. 1 carbon, 2 hydrogens, 1 oxygen. And when we look at the subscript outside of the parentheses, that tells us how many carbons are present. So when you write the equation CH2O with the subscript 6 outside, which specific carbohydrate are we talking about that has 6 carbons? Glucose. So you can write it that way, or you can write it C6H12O6. It means the same thing. So in our equation, and starting on, let's go ahead and start on the right side. Because this is photosynthesis. With photosynthesis, we're going to take 6 molecules of carbon dioxide. And as we talk about balancing the equation, when we have 6 molecules of CO2, how many carbons do we have? 6 molecules of CO2, how many molecules of carbon are there? 6. So photosynthesis is going to take the carbons in these 6 individual molecules, and it's going to link them together into one 6-carbon molecule called glucose. That's really what photosynthesis is about. And the energy is going to come from where? The sun. Because where does all energy come from initially anyway? The sun. And that's how plants do that. So in our equation, plants take carbon dioxide, they take water, and the energy that we have designated here is coming from the sun. And using that energy, they assimilate these carbons into one molecule called glucose. Now, when you have an equation, and we see the arrow pointing away from the side where you see energy. So in this case, if the arrow is pointing toward the left, and energy is on the right, meaning energy is there before and must be there so the reaction can occur. What would energy be in this case, a reactant or a product? It's a reactant. And so to have this chemical reaction take place, you have to put energy into the system. That kind of equation is called an endergonic. Used to be called endothermic, but now the term is endergonic reaction, meaning you've got to capture and use energy to make that reaction happen. So photosynthesis, energy in, so that we can take our carbons and link them together into one molecule called glucose. Now remember, that energy transfer is not going to be 100% efficient. You're going to lose some of that energy in the transfer. Which law of thermodynamics states that? Second. You had a 50-50 shot, right? There's only 2. It's the second. The first one says you can't make something from nothing. So here we've got our energy in, and the energy from the sun is captured and stored in the chemical bonds of glucose. Now, we're going to go over, and that happens in our chloroplasts, by the way, in our plants. Now we're going to take the glucose, and we are going to begin to process the glucose in our cells. And the mitochondria, in addition to some other enzymes that are happening in the cytoplasm, are going to begin to break down the glucose and recover the energy. And that's our reaction here. We see our glucose. We're going to have to use oxygen. So this is aerobic respiration. Get familiar with that term, aerobic. You see those are both to the left of the arrow. Arrow's moving toward the right. So these are now our reactants. And after aerobic respiration, we are going to break the glucose down into what? 6 molecules of carbon dioxide. We're going to produce 6 molecules of water. And what is being released? Energy. Do you see how the equations are practically the same? What's the really only difference? The direction of the arrow. So if you remember the components of the equation and you know that an endergonic, putting energy in, the arrow's pointing away from the energy. And when the arrow points towards the release or the production of energy, that is called exergonic or exothermic. So these terms really do strictly point toward, are you having to use energy for the reaction to occur, or are you releasing the energy because the reaction occurred? Plants store the energy. Mitochondria release it. Now, why didn't I say plants store it, animals release it? Because guess what? Plants have, in addition to chloroplasts, they have mitochondria as well. All cells are going to have that. They have that for the energy production. So do we understand our balanced equation? Energy in, endergonic, energy out, exergonic. That's just sort of foundational to where we're going. Because to understand how we can build carbon dioxide into glucose and tear glucose down into carbon dioxide, it's not enough to understand the chemical reactions, because these will not occur efficiently enough for life to exist without these all-important proteins called enzymes. Now, first of all, let me be very clear. Enzymes are not magicians. You know, magicians appear to make something happen that shouldn't happen. The one that I'm most fascinated by are the magicians that appear to levitate off the ground. Have you ever seen those? That freaks me out. I wish somebody would tell me how they do that. Enzymes aren't magicians. They're not going to make something happen that couldn't normally happen. All enzymes can do is take a naturally occurring reaction and help it happen easier. And because it's happening easier, it's going to happen faster. That's all enzymes do. They're not going to make a reaction happen that wouldn't normally happen in nature. And so we say enzymes are catalysts in that case. They facilitate naturally occurring reactions. They increase the rate of naturally occurring reactions. But you see, the key to all this is the reactions are already going to happen. Enzymes just help that. So if we look at a chart, here are our reactants, and this is sort of energy. So here are our reactants. Here's our products. Notice the energy of our products is less than the energy of our reactants, and so energy's being released, energy's being given off. What kind of reaction did we just label in the last slide means energy given up? Exergonic. You know one way I remember that? The first part of exergonic, ex, sounds like exit, which is to leave. So if you remember exit, leave, exergonic energy leaving, then the other one has to be energy in, right? So that's how you can remember that. But notice, it's not just a straight hill down. You've got this big hump. You actually have to put energy into the system before you can release the energy. And this additional energy up is called the activation energy for the reaction to occur. Now, that is an uncatalyzed reaction that you're looking at. That's what naturally occurring reactions would have to do. Many of them would have to put that energy in, and a substantial bit of energy, before you could start downhill and get to the products. So the way enzymes function as catalysts is they reduce this activation energy. Now, in this illustration, you see the blue line that has the higher hump. That's the higher activation energy. That is an uncatalyzed reaction. No enzymes present. The red line that is a much shorter hill to get over, much lower activation energy, that is a catalyzed reaction because we put enzymes in. Now, let's go back to our example of hydrogen peroxide. You remember what hydrogen peroxide broke down into in that other very simple equation? It broke down into oxygen and water. So if we just follow an uncatalyzed reaction, if we just put hydrogen peroxide in a beaker, set it on the counter in your bathroom, is that really going to effectively change into water and oxygen? Everybody shake your head no. To add the energy, to get up that activation energy hill, you'd have to heat it up, put energy in, right? But if you add some hydrolase enzyme and you leave that beaker at room temperature, that's what's going to happen. Why does it bubble up like that? What are you producing? Oxygen. You're producing oxygen. It's a gas. So it's going to come out of solution. Do you see the difference? This is uncatalyzed, no enzyme. It will happen eventually if you give it some energy. But if you don't have that much energy around, but you've got some enzymes, boom, you can make it go, and you can make it go a lot faster and a lot more efficiently. That's the use of enzymes. Now, there's more than just one enzyme in the world. There's many different categories of enzymes. We're going to give you 5 categories in a bit. But what I want you to understand about enzymes is, in addition to the fact that the majority of enzymes are proteins, now there are a few that are made of RNA, and we're really not going to get into those. For our purposes here, all enzymes are proteins. Is that good? And so proteins are going to have a very specific shape, right? We talked about that. You know our 4 levels of protein organization. I mean, you can follow the numbers: primary, secondary, tertiary, quaternary. So with the specific shape of our proteins called enzymes, part of that means is a specific shape that is going to bind to one of our reactants in our chemical equation. And we changed the name a little bit for those reactants, because if something is going to bind to an enzyme, we don't necessarily call it a reactant. Now we call it a substrate. But it's still the same thing as a reactant. It's what happens before the reaction occurs. But because of the shape of our enzyme, there is a specific pocket that is going to be just the right shape for only one specific substrate. And that pocket is called the active site. Now, many of us in here have cars. And many of us, well, all of us that have cars, have a car key. But is your car key going to fit her car? No. It's not a specific match. So not every substrate can bind to the active site of just any enzyme. It is going to be specific. There used to be a lock and key sort of analogy to enzymes, and I still like to think of that as far as binding of the substrate to the active site where that material is going to be processed. Broken down, built up, some change is going to happen because of that active site and because of the enzyme. But it's more than just a specific fit. Because what we understand now is that when you have an enzyme that has a very specific active site, when the substrate, as you can see in the lower right, when it binds to the enzyme changes shape. Remember, we said a lot of those hydrogen bonds are there to provide the flexibility for the enzyme. And as the enzyme changes shape because the substrate bound, do you see how we've stretched out the substrate itself? And now we've made it a lot easier for the enzyme in this particular illustration to cut that substrate into 2 separate pieces. So instead of a lock and key, now we refer to this as the induced fit model. There's a fit between the substrate and the active site, and the binding induces the enzyme and the substrate that is now bound to the enzyme to change shape. And it's that change of shape that lowers the activation energy to make the equation, to make the chemical reaction easier to happen. And in many instances, the active site of our enzyme is going to have very specific amino acids with very specific properties. And it's because of those important amino acids in that active site, that's what facilitates the binding and the change of the conformation over the entire protein called the enzyme. Now, understand this word conformation. This means shape. When you change conformation, that means change shape. It's just a big fancy scientific word for that. So make sure you note that, because you'll hear that quite a bit in biochemistry stuff and anatomy stuff, especially as it relates to enzymes. Now, there are 6 categories of enzymes. We're not going to learn specific examples of all of these. I simply want you to be familiar with these 6 categories in a very fundamental sense, because there are a lot of different enzymes and a lot of different ways to characterize enzymes. The first thing you're going to notice about enzymes, when you hear the name of an enzyme and this word ends with ASE, ACE, your little warning light should be going off. Okay, this is an enzyme. What does it do? And typically the first part of the name tells you what it does. And many enzymes are catabolic. They break things down. So if I were to tell you there was an enzyme that was called DNase, what do you think that enzyme does? Breaks down DNA. What about RNase? Breaks down RNA. What about a protease? Breaks down protein. What about a lipase? Breaks down lipid. You see, that's fairly straightforward. But then we get into some of these other categories. Oxidoreductase. Well, these enzymes are going to use oxidation reduction reactions, adding or taking away electrons to change molecules. Transferases. We're going to transfer atoms or molecules between structures. Hydrolases, you're just going to cut a molecule into 2 pieces like we saw in our illustration. An isomerase is going to change a molecule such that it's going to look like the mirror image of itself. And when I talk about mirror image, are your hands identical? Okay, we're not talking about fingerprints. But are your hands identical? No. Because this hand's got the thumb pointing toward the right and this hand's got the thumb. You see how they're mirror images though? These would be isomers. Your hands are isomers of each other. And these enzymes are good at taking a molecule and flipping parts to present a mirror image copy. That's an isomerase. A ligase, bless you. A ligase links 2 molecules together. DNA ligase links 2 molecules of what together? DNA. That's going to be an important part when we get to cellular replication and DNA replication. That's a critical part of your cells dividing. And then finally, these lyases, this is a removal of a specific group. Now there's some overlap to some of these. Some of these sound similar. So don't panic if you don't know a specific difference between these. I mainly want you to be familiar with the names of these 6 categories. And again, this is just foundational stuff that you may run into later. But it's important to understand the diversity of enzymes, just like, man, there's an extreme diversity of proteins. All right. Now, this is a chart. I know you can't read it. The words are small. This is a chart that, again, shows those 6 categories, gives you some examples, gives you a little bit of more explanation, and shows actual chemical reactions that these enzymes facilitate. Use this chart only if it helps you. If this chart only, if you're looking at it and it starts to confuse you or starts to make you anxious, don't look at it. I'm not asking you to memorize this chart. Is that clear? This is just help you to understand, if you wish to, better those 6 categories of enzymes. Now, what's more important necessarily than just the categories is how do we regulate enzymes? They're facilitating chemical reactions. That's going to be a critical aspect of maintaining our balance for our cells and our tissues, our organs and organ systems. And what's that big word that we have to attain balance? What is that word? Homeostasis. So enzymes are a part of that. And regulating how fast or how slow they facilitate these reactions, or if they do them or if they don't, that's a huge part of this. So we're going to step through these 5 means with which we regulate enzyme activity and the speed with which they in fact facilitate these reactions. So first, substrate level regulation. What is another word for a substrate? What did we say a substrate was? A reactant. The substrate is a reactant. So this is a little kind of changing our perspective. We've been focused on the enzyme. Well, now we're going to focus on the substrate. But do you see how that can impact a rate of reaction? If you have a ton of enzyme and no substrate, is the reaction going to happen? No, it's not. But I think you can also see as you increase the concentration of substrate, not increasing the number of enzymes, you can see how our reaction rate increases. I mean, that's just logic, right? But you notice we get to a certain concentration and the enzymes can't work any faster. You've saturated the enzymes. The substrate starts to stack up. And sometimes this is a difficult concept, so I enlisted some old television show to help with this. So imagine Lucy and Ethel are the enzymes. The reaction that they're facilitating is wrapping the candy. Have you seen this clip before? The belt starts going faster, and it starts going faster, and they're not able to handle that amount. They're still only wrapping a certain amount, even though there's more and more and more. But does that make sense, though? As you get more and more substrate, the substrate has to line up and wait because the reactions, the enzymes can't make it go any faster. And that's why you see that plateau. Now, if you did have a ton of substrate and you reach that plateau of enzyme activity, but your cells or your body needed more product, what is something the cell could do to increase the rate? Think about it logically. If you need to increase the rate, but the number of enzymes you have are working as hard as they can, how can you make it go faster? Make more enzymes. Make more enzymes. So that's another way the cell can enhance its activity. If it needs more enzyme and you've got plenty of substrate, the cell can always make more enzyme and make the process go faster, produce more product more quickly. Now, another step of regulation is temperature.